Thanks, Dan, and thanks for the opportunity to be face-to-face. It's been a little while since that's happened. I think last time I spoke to you was about gene editing at one of these meetings we had in Dunedin 2 or 3 years ago. So over the last year or two, I've had a number of people saying to me, we're at a bit of a crossroads with wool, and we can keep producing it, but often it's costing us more to remove it than what we're getting paid for it. Well, there are two other options other than continuing along the status quo. One is to go to shedding sheep, and the other one is to go to a finer wooled sheep. And my response to that is we've got some really good tools for doing that now, and it's called introgression. And I got some blank looks on my face. What do you mean about introgression? So I guess in a weak moment, I agreed to talk to you about genomic introgression, and I think Becky might have said it was a future tool for breeders, but after listening to your discussions and seeing the phone some of you had, maybe it should be a tool for future breeders, because the technologies I'm talking about, you're really going to need some devices like phones or computers to implement some of these things. So genetic improvement in sheep and beef cattle, usually what we're doing is making a stepwise change. And they're only little steps of the order of 1% is kind of what we think is a normal ballpark. And it doesn't sound like much making a 1% change, but after say 20 years or 40 years, the length of time I've been in the business at Massey, you know, that's a 40% improvement in whatever was happening before. So every year we're trying to make the new crop of lambs or calves a little bit better than they were before. And that's quite different from what they tend to do in many plant industries. In many plant breeding industries, it's a bit more like cars or trucks. Behind the scenes, they have a whole lot of R&D trying to make things better all the time. But what they release on the market is a new variety. And the new variety often has to be quite different from the ones that existed in the past. And quite often, at least in examples like maize and corn, those seeds they sell are F1 seeds. So they're all genetically identical, and their challenge each year is to come up with a new F1 seed that was better than the one that they had last year. And one of the ways they do that is with introgression, because they have new disease problems which appear every year. So one of the things we know is you could have a high-performing sheep or a high-performing cow or a high-performing corn plant, but it won't perform highly if you have a problem, like if you run out of feed, or it's really cold, or it's really wet, or you have a disease problem. So in the plant industry, they have produced GM varieties. So they've done things like GM resistance, glyphosate resistant. So they've introduced genes into maize to make them resistant. They've also put them into other varieties, other species. They've made drought-resistant plants through GM, and they've made what's known as BT corn. And BT corn is something where they've introduced something from Bacillus thuringiensis, which produces spores that kill off some of the disease problems. So they've got a whole lot of these GM varieties that have one of these attributes. They refer to them as traits. I don't really like that word, but they refer to them as traits. So what they want to do now is to take a high-yielding potential plant and stick all these other things into it. And they refer to that as stacking the traits or stacking the genes. And they do that by introgression. So they do it by old-fashioned crossing, because if they do it with old-fashioned crossing, then they can end up with a high-yielding realization, because the high-yielding variety now has protection from all these other things, and they don't need any new GM licensing, because the GM licensing was already done for each of those varieties when they had one of these attributes. An example of one of those things would be smart stacks. And just like the concept you've heard about refugia in your worm selection, they do the same kind of things there. So they mix up some seed that is refuge seed so that they don't get too active selection for these things that they're trying to protect against. Okay, so the next topic I want to talk about is true breeding. So true breeding means that the offspring will carry the same or a similar phenotype as the parents do. And most breed attributes, most things that you think about when you see an animal and recognize its breed, is something that is usually meant to be true breeding. So they are things that were typically mandatory for pedigree registration. For instance, Angus cattle are black and Angus cattle are polled. It's a requirement of the registration process, right? If you saw a black animal that was horned, you wouldn't immediately think it was Angus, as an example. Or if it was red and white colour, you might recognise it as a Hereford. So one of the issues is that you can't actually produce a breed. To produce a breed, it's like producing a new t-shirt or a new pair of shoes or a new phone. It's got to be recognisable. You've got to stimulate demand for it. Everybody has to recognise that you've got it and it's different from what everybody else has got. So if you're going to produce a recognisable breed, you have to put in some true breeding characteristic. And if you think about what are some of the new breeds that have come along recently, at least in the cattle area, Highland cattle. Why were those so interesting to people? Because they looked different from all the other breeds that were already out there. Another one would be the speckled park, right? And an old-fashioned one would have been the belted Galloway. So people like having those kind of breeds because when you drive along the road, you look out and you can see them a little bit different. But I remember some years ago driving along with the late Holmes Warren in a car, and we were driving past some sheep, and he looked out the window and he said, that must be one of your client, Derek. And speaking to Derek Daniel, and it always amazed me that just driving along at high speed, that Holmes was able to recognise some of those true breeding characteristics, that those were Wairiri sheep in that paddock and not just some other kind of Romney. Most of my students would struggle to tell the difference between a Romney and many other breeds. So many examples of true breeding characteristics that are important for breed definition. But in addition, sometimes we have what we know of as complex traits, which are responsible for breed differences. And complex traits are things that have got usually many genes involved. The inheritance of them is much more complicated. But if you make the animals extremely different enough, then they can still be part of the breed attribute. So an example would be marbling and Wagyu. You know, marbling and Wagyu is not controlled by one major gene, it's controlled lots of genes. But marbling and Wagyu is so different from marbling and most other breeds that it works as a breed attribute. Okay, so for management and marketing purposes, we want animals to be somewhat uniform and we want them to be true breeding. So for dual purpose sheep breeds, we usually like them to have white wool, right? It was a real pain when you had a black sheep turn up in your flock, as would happen from time to time. For merino sheep, we want them to have soft wool that's fine enough to get a premium. And ideally, we want it to be all over quite a narrow range of fineness. And if we have shedding sheep, we want them all to shed. We don't want to have to get a shearer in to shear the handful of them that don't shed. So that's most easily achieved if the characteristic we're interested in introgressing is controlled by one gene we call monogenic, or a few genes, we talk about oligogenic, or the worst case would be lots of genes, which we talk about as being polygenic. Okay, so when I looked at my slides last night, I thought, what was I thinking, deciding to try and talk to you about this? So I'm just going to have to try and walk you through it and bear with me. So, mammals and many animals are diploid, which means their chromosomes are in pairs. They get one member of the pair from the father and one member of their pair from the mother. So here I'm showing you three individuals, and I'm showing you here, I've painted the blue chromosome here for the paternal one and the green chromosome for the maternal one. So this might be a normal individual, and I've used a plus, meaning it's wild type, which is just genetics jargon for meaning the normal kind of animal that you would see. And then we might have some other kind of individual which similarly has a chromosome came from its father and one that came from its mother, but it's got some major gene effect I'm representing by the letter M, which is different from the wild type. And we would talk about these individuals here as being homozygous because when they produce zygotes, like sperm or eggs, they're all the same, which is homo means the same zygote because they all carry the plus. And these ones here would also be homozygous because their sperm or eggs would all carry the capital M. But if we were to cross those, say, we could get an individual like this, which would be a heterozygote, and half of its gametes would carry the plus and half would carry the M, and that would cause the phenotype to segregate and we'd see variation in it, which is something we don't want if we want a true breeding attribute. Okay. So, some of the history of this kind of technology goes back to a very interesting character, and I never met him myself, but this is Dr. Dry, Daddy Dry as he was known of. He referred to his wife as Mammy, and I'm not quite sure when he got the name Daddy, but people didn't refer to him to his face, but Daddy Dry, when he came to New Zealand, had been working on Wensleydales. So for those of you whose kids are into Wallace and Gromit, you know, Wensleydales really are a breed. And one of the interesting things that Dr. Dry found about Wensleydales was the type of Wensleydale that met the kind the Breeds Society book said you should have was actually enriched in heterozygotes, which means they wouldn't be true breeding, which seems like a bad thing to have in a breed standard. The other interesting thing that he showed was that actually black sheep in Wensleydale was due to a recessive gene. So when you had two copies of the black, it ended up being black. And if you had one copy of black and one copy of wild type, it would be white. So black could keep turning up in the future. So he was the first person to show that many, many years ago. And when he came to New Zealand at Massey, he was interested in hair, which is when the wool is hollow. It's different from a wool fibre itself. And one of the genes that he found responsible for that, what he called the N gene, named after a farmer, Nielsen, is what led to the Drysdale when it was introgressed into cheviot animals. But the other remarkable thing about Dr. Dry is he showed that if you looked at the mid-side, that's just in here under the shoulder, although it was a dominant gene, which meant that one copy of the N gene produced a hairy animal, just like two copies of them did, right on the birth coat you could distinguish them. So you could tell who was a carrier, and you could use that for selection. And he used that to his advantage to produce Drysdales much faster than you otherwise could do. So now I want you to think about the problem that I mentioned right at the beginning, which is where we have some wild type breed in New Zealand that would most commonly be, say, Romneys, and there's some other breed that's got some attribute that you have now decided is desirable. So it might be a Merino because it's got fine wool if you want to go down that route, or it might be a Wiltshire because it sheds because you want to go down that route, or it could be some other completely different attribute that you want to bring into your breed that doesn't currently exist. And there are lots of examples of things that we might want to bring in. And I think as an industry, you guys haven't been very good about robbing good genes from other breeds and bringing them into your own breeds. And generally, if you're too breed association centric, then you don't like the idea of bringing something in from outside. But if you don't have variation for the thing you want to change in your own breed, then it's very hard to make progress by selection. So one option you could do is grading up. And grading up means that whatever you're changing to is presumably better than what you started with. So if you were to grade up to something like a Merino or a Wiltshire, then you would start with your wild type, and now I'm using the colour of the chromosomes to label the breed they came from. So you're starting with blue ones that have got pluses, and you're ending up with orange ones that came from another breed that have got, say, M's. And if you were going to do that, when you eventually get there, the old breed has completely gone. And you'd have to hope that the new breed really is better than the old breed, and you need to think about that in a pasture-based whole system context. So if you wanted to go full from your Romneys to Merinos, then you'd have to hope that those Merinos are better, and hope that your ancestors here that ran these things in some of the North Island many, many years ago who decided they weren't good had somehow got it wrong, for instance. Okay, the other thing that you might want to do, if you don't believe that replacing the breed would be a better proposition, would be to backcross. And so I mentioned earlier at the beginning that selection is about making these small incremental changes. So there have been small incremental changes been going on in breeds like Romneys since the 1960s at least, for New Zealand circumstances, right? So we've got a huge amount of selection for facial eczema, worm resistance, all kinds of things. If I want to bring in fine wool or I want to bring in shedding, I don't want to give up all of that selection that's occurred in the Romney. So instead what I might want to do is backcross my plus animals and then do some selection for a number of generations until I end up with individuals whose chromosomes are just like they were in my original breed, except I've changed the gene or genes for this true breeding attribute. Okay, so whether I'm grading up or whether I am backcrossing, the first mating would be the same. I take one of my founder breeds, the blue one with the pluses, and I take the other breed with the major gene effect M, and I cross them. And the only kind of gamete I can get from the blue breed carries the whole blue copy of the chromosome, because that's the only copy of chromosomes they have. And the orange breed carries the orange chromosome. So the first cross animals are all going to have one member of each pair having a blue origin and one having an orange origin. And one of the problems we see when people do this is these animals often exhibit quite a lot of heterosis. And farmers think, wow, that's an amazing improvement on what I had before. And so then they decide they should keep on going and grading up to this, and then they find that they end up grading back down. And a classic case would be when the Holstein first came into New Zealand, got mated to Friesians. The Holstein Friesians produced a lot of milk. The reproduction was okay. They mated them to Holsteins again. Their milk production was even better, and their reproduction was a whole lot worse. And as the Irish showed in a whole system context, they were much less profitable. So you've got to be wary about what happens in that first cross. Okay, but then what happens in the next cross is if you take that F1 parent and you get gametes from the F1 parent, then about 20% of the time, what you're going to get in the sperm or the egg is an intact copy of that chromosome. So you're going to get a blue chromosome or you're going to get an orange chromosome. It's not exactly 20% because it varies a bit from chromosome to chromosome, depending on how long the chromosome is. But this is a trick that they use widely in genetics experiments with things like mice to produce what they call consomic lines. So in mice, they try and produce a line of mice where all of the 20 pairs of chromosomes except one come from one strain and the other chromosome comes from the other strain. So they can test the effect of every one of the chromosomes independently. So about 20% of the time, roughly, that's what you'd see, at least that's what I see when I look at this data in cattle. About 2/3 of the time, you'll get a recombination event. And a recombination event occurs when that blue and that orange chromosome tangle up and they exchange. And they exchange at some particular crossover point. And I've shown it, say here, this is a crossover point. So most of the chromosome inherited the blue kind, which had the plus inside it. And then there was a crossover, and the tail of this chromosome is the orange kind. And at that same recombination event, we could conceptually generate the opposite kind of chromosome, which is mostly orange and a little bit blue. So most of the gametes that are produced from an F1 will be like this. There'll be a mixture of chromosomes that came from the two original breeds, and the crossover could more or less occur anywhere along the chromosome from one end to the other. Although there are some points, what we call recombination hotspots, where they're more likely to cross over and some where they're less likely to cross over. Occasionally, what we'll see is a double crossover where there's two recombinations. So for instance, a recombination right down this end, so the beginning might be blue, then there's a big chunk of orange and then another blue bit, or there's the reverse kind there. When this happens, the two crossovers don't occur close together, there's interference. So when this happens, it's usually they're kind of distant, at least half a chromosome apart. Okay, so let's think about this now. If we're going to grade up, and we might do this without selection, or we might do it with selection. So from a dual-purpose Romney to, say, a Merino or a Wiltshire. So in the F1 parent, we'll generate a chromosome with the mixed colour, like the bottom one there. But if I'm grading up, I'm mating every generation back to that other breed, the orange breed. So one of the chromosomes is going to be totally orange, and the other chromosome is going to be a mixture of blue and orange. And in the first generation in those F1s, they're going to be half orange and half blue, and half of them are going to have one copy of that big M. And the next generation, they're going to be 3/4 orange, and 3/4 of them are going to have a copy of the big M. And if I keep on doing this, then they'll end up 7/8, 15/16, 31/62s, and 63/64. And if you want to have a big argument, you ask a breed association how many generations of backcrossing are required before you can register as an animal in that breed. But it doesn't matter how many generations you go, you'll never get to 100%. But as time goes on, they'll get more and more orange and there'll be less and less blue. But if we stop after we've got to, say, 63/64, then the animals, 1 in 64 of them, will still carry that plus allele if we've done no selection for M. And if we were mating randomly, then 1/64 of the rams and 1 out of 64 of the ewes will carry the plus over. So 1 in 4,096 matings will be between two carriers. And a carrier-carrier mating has a 1 in 4 chance that you'll generate a plus phenotype. So that means something like 1 in 16,000 lambs, you'll see that off type. And that's why, you see these black sheep popping up in commercial flocks of white sheep, even though the breeder thinks they have been getting rid of everything that has had black in it. Okay, and usually we only hear about this in commercial flocks. So most of the genetic defects that I have ever found, and I've found a number in sheep, have always come to me when a commercial farmer has contacted their vet about a strange animal, and we've gone back and we've found a defect, and eventually we trace it back to the ram breeding flock. And often the ram breeder admits that, yes, we saw a few of those animals years ago and we got rid of every sire and dam that produced them and thought we'd cleaned it out. But when you've got lots of commercial clients, they will pop up out there. So if you can do selection for that M, if you can tell the animals that have got the M, then you can do a much better job. So you can't do anything in the F1 because they're half orange and only half of them are M. But you could make sure that the only individuals you keep when you mate those to each others are the ones that are big M. So they would be true breeding right from that 3/4 point. And each year as you go through, you can reduce the amount of orange, but you'll still end up with them being homozygous for the M. So they'll be true breeding for the thing you just introgressed, but they're still going to have a little bit of that blue background which is still going to pop up a little bit from time to time. So things like if you've got descendants from merinos and you've done all this, you'll still get foot rot problems and pink noses and other things popping up year after year after year. It's very, very hard to get rid of them. And each generation you go on with the backcross, you break those fragments down to smaller and smaller pieces, which makes it harder and harder to get rid of them. So the other option, backcrossing with selection. So for instance, from Romney to a fine wool Romney or a shedding Romney. So same kind of concept again, where we're starting off at the program with an F1, with one of the blues with a plus and one of the oranges with an M. And what we want to end up at the end is the blue, our original Romney background, but we want to have introgressed in this big M piece. And how easy this is depends on whether we can distinguish the M plus individuals from the plus pluses or not. And what was a revolution for the Drysdale was Dr. Dry discovering that you could do that on the birth coat of those lambs. Otherwise, you would have had normal woolly Romneys popping up amongst Drysdales all the time. But he was able to purge those out pretty quickly. So if we can select on the M plus, then it's pretty simple to introgress the M allele from some other breed. So I take the F1s. These are 8 different offspring I might generate when I mate the F1 backcross to my original blue type. So every one of these 8 has a blue chromosome that carries the plus from my backcross to, say, the Romney, but the other chromosome will be a mixture of orange and blue. And if I'm only keeping the ones with M, then those are kind of the 8 offspring that I would allow to go forward. Any individuals from the F1 parent that had a plus here would be a plus plus. If I can distinguish those, I can get rid of them and not use them as a parent. But when I do this, one thing you'll notice is it's going to have orange kind of around that M locus. So after we've done this for a long, long time, the amount of M here will reduce, but it will still leave what we know, what we refer to as a selection signature. So that's why we can look at a breed now and we can tell how something was introgressed into it, even though that introgression may have been thousands of years ago, because there's a selection signature. Okay, so in this particular case, I start off in my F1 and they're half orange. And the next generation, the first backcross generation, they're going to go to quarter orange, then they're going to go to 8th orange, then they go to 16th orange, and then they're going to go to 1/32 orange. And at the end of my program, you can see there's a bit of an orange selection signature sitting around that gene that I've introgressed. And there's also other little wee bits of orange sitting up there, right? Because if I've still got 1/32 orange or 1/64 orange, there's still going to be a few little orange bits sitting up there. And that's exactly the case now for most of us in this room. We've got little fragments of Neanderthal and we've got little fragments of Denisovan that come back from kind of these introgression events that occurred a long, long time ago. But at some point, these individuals here, these backcrosses, they're all heterozygous. And if you want them to be true breeding, you're going to have to mate the heterozygote to the heterozygote and keep the animals that are MM before you make them true breeding. So if you do that too early on, then you're going to have a lot of that orange stuff floating around. So the longer you wait, the better you'll be able to get rid of that other background, but the longer you're going to have to wait before you get those true breeding double M's. Okay. The other option is where instead of being able to recognize the M plus, it may be that the M plus isn't different enough from the plus plus for you to identify who is a carrier. And that means that you're going to have to do the selection based on the MMs. So the way that would work is you would make an F1, so it's carrying, every individual's carrying one copy of M and one carrier of plus, and then you'd mate those F1s to themselves, and you would have a quarter of them that are big M, a quarter of them would be plus plus, and half of them would be M plus. So you would keep just those quarter that were like this, but they would have half of their background would be orange, which might be the breed you don't want. So you could then backcross it again to blue to knock the orange from half down to a quarter, but now they are M plus again. So then you're going to have to make them to themselves again, and then you're going to have quarter orange that are all MM, and then you're going to go to eighth and so on. So a much more complicated process. So this just kind of demonstrates what it looks like. So here we're going the two ways, at the top when we can identify M plus, and the bottom when we can only identify MM. So this is generation 1, 2, 3, 4, 5, 6. So 6 generations, about a 3-year generation interval roughly in sheep, depending on how you do it. So it's going to take you 15 to 20 years to get to this point out here. And if you can identify the M plus, you'll only have 1/32 of the genome still adulterated from the breed you stole the favourable gene from. If you've had to do it this way, then you're going to have 1/8 of it. And I think that's probably the kind of thing that has happened when people have tried to introgress fine wool from merinos into Romneys, and they've ended up still with a lot of the unfavourable merino characteristics for the kind of country that you're trying to run these half-breds on. Okay, so introgressing, intercrossing is about stabilising a crossbred, and that's the result of interbreeding these crosses. And when you remember, when we're backcrossing or grading up, one chromosome comes totally from one breed and the other one is a mix. When we stabilise, what's going to happen is both chromosomes are going to be a mixture of both of the background. And usually it's done, so a simple inherited characteristic will be true breeding. So for instance, it's what Leachman did to create the breeds that became known as stabiliser, right? He was stabilising it because if he followed the recipe that USDA Meat Animal Research Centre suggested for creating F1 crosses of beef cattle, you ended up with a whole bunch of different coat colours. So he decided to stabilise stabilise the breed. Okay, and so if stabilisation occurs when you say quarter orange, then those breed percentages will remain around. The chunks of orange will get smaller and smaller, which means they'll get harder and harder to get rid of because they'll be spread everywhere throughout the genome, but they will still hang around there unless they happen to be favourable or unfavourable and you're really good at doing selection for them. Then you may be able to get rid of them from certain regions. And that's the kind of thing that's happened with Neanderthal and Denisovan. They're not rampant across the whole genome. They are more common in particular areas where they had some advantage. So how can genomics help with all this? Well, if you can directly test for the thing you want, then you can tell who's heterozygous and tell who is homozygous, and it's a revolutionary improvement. And that's particularly true when you can't look at the phenotype and tell what the genotype is. And some examples of that are going on in New Zealand at the moment are the slick gene variant, which has been borrowed or stolen from the Senapole. And the problem is that the slick hair coat doesn't occur really until they're later in age, until they've been well fed and until it's hot. It's very hard to phenotype them accurately. Another example would be polled. There's 4 different alleles affecting polled. The 2 common ones are Frisian and Celtic. And phenotype to recognise whether animals are horned or polled is not that easy because of problems with skurs. And one of the reasons it took so long to find the mutations responsible for polled and horned was because farmers making occasional mistake in their phenotypes. Okay, so unlike slick horns or say the A2 beta casein that you might hear a lot about, in most cases the genomic location of the genes we want aren't known. The number of genes involved might be unknown, the specific of genes might not be known, the particular variant may not be known, and maybe even the gross location, or what we call the QTL, may be unknown. So some relevant sheep examples would be the genes in merinos that would confer fine, heavy fleeces. We don't know exactly what those are. But one of the things we know is that primary wool follicles are arranged in threes with sweat glands and erector muscles, and then there are secondary fibres around those. And in a Romney kind of breed, the secondary to primary ratio is typically around 3 to 5. So if you select for fineness, what tends to happen is you get a collapse in fleece weight. So if you want a heavy fleece, fine wool individual, you need to increase the number of primary follicles and you need to increase the ratio of secondary to primary follicles. But there's not much variation, genetic variation in the ratio of secondary to primary follicles in Romneys. So you really have to do some introgression if you want to do it. Aseasonality would be another one that would be great to have, and shedding would be another one. They'd all be good things to introgress. So in order to do it, what we need to do is to phase the genotypes. So when you send a sample off and get it genotyped, what comes back is a call which we can transfer into a number like 0, 1 or 2 copies of a particular allele. So an individual might, this is 4 different loci or locations on the genome, and their genotype is 2, 0, 1 or 1. Then what I want to know is what's the paternal haplotype, what's the chromosome that came from the father, and what's the chromosome that came from the mother on that. And if the individual has 2, I know he must have got a 1 from both his father and his mother. If he's got a 0, I know he must have got a 0 from both his father and mother. But if he got a 1, he's got a 0 from one parent and a 1 from the other. So he could have had this particular phase, or this phase, or this phase, or this phase would all give the same genotype. So in order to use the genomics, we have to phase them. So this is a real life example from a bull that I had to work on recently. And I've just pulled out the loci where he was heterozygous. So the bull himself there, you see, is all 1s. And there's his sire and his dam and 2 of his offspring. That's the raw SNP data. The first thing we do is phase the homozygote. So whenever I have a 0 in the genotype, I say both the paternal and maternal must be 0. Wherever I have a 2, both the paternal and the maternal must be a 1. And when the individual is a heterozygous, I can't tell. So I do that, and then I drop the known phase from the parents to the offspring. So you can see up there what I was able to resolve from the father. I know that must have been passed on as the paternal haplotype, which I've shown in orange to the sire, and the maternal haplotype, what I've shown in blue there for the dam. And you can see in some positions, I know the bull is heterozygote, the mother gave a 0, the father gave a question mark. I can resolve the question mark must have been a 1 because we know he's heterozygous. So we can fill all those things in, and then that gives me a lot more information, and then I fill in by looking from the father down to his offspring. And by the time, and then I reconcile the parent and offspring haplotypes, and then when I'm done, I get kind of data like this. So now I've actually been able to colour these things. So a particular fragment of the genome, this is the first chunk of chromosome 7, I can tell you exactly where it came from. So I can track all the way back to the founder, say Romney and Merino, or the founder Romney and Wiltshire. I can tell you what's a Wiltshire fragment and what's a Merino fragment or a Romney fragment. And that means that I can do some interesting things. It allows me to do things like compare the offspring that got a particular fragment from Wiltshire with the offspring of the same sire that got the corresponding fragment from the Romney. And if they differ for shedding, then I know that Wiltshire chunk had something important for shedding, and I can select to bring it in. And I'm going to have to genotype over subsequent generations, and I may have to do phenotyping. I might not have to do phenotyping. It just depends. And for some traits, like SP ratio, phenotyping is going to be difficult and expensive. For other, like shedding and Wiltshires, it's going to be very easy. You can just do it by eye. So several benefits from the phasing. We can identify the fragments contributing to the favoured phenotype, like the shedding in the Wiltshire, and we can then use that knowledge to select descendants that have the desirable fragment, but I can also use that to cull the descendants that carry fragments that I know are not useful. So remember, I want to keep all the Fe resistance and all the worm resistance and the ability to count that those Romney-Us have. I just want to bring in the shedding genes. And I can also use it to choose matings between different sets of individuals that carry favourable fragments. I want to be greedy. I want to produce eventually an introgressed breed that is true breeding for every desirable fragment. And some of the individuals will be desirable at one place, some will be desirable at the other. I might have to cross them to generate some offspring that are desirable in both places. Okay, so problems in practice. The desired phenotype may not be controlled by just one gene, and that means that some animals with the desired phenotype may not be true breeding. That's going to make it a bit harder. Selection might need to simultaneously address more than one of these QTL. Larger discovery populations will be needed. And the worst case scenario is the phenotype represents a complex trait and there's lots of genes for small effects and they're all over the place. I can still do it, but it will be a bit harder, and it just means that larger populations will be required. So summary, finally, for those of you flashing red lights at me. Populations can be slow and systematically improved, say 1% a year by within population selection, provided genetic variation already exists in that trait, in that population. Desirable attributes from one population can be introduced into another in a quantum leap. The best quantum leap would be through gene editing, but we'd have to know the actual mutation and we'd have to be allowed to do it. Otherwise, we can do it this conventional way known as introgression, and we can do that to retain the advantages of the original population. It's much easier to achieve with genomics than using phenotypes, and it will benefit from using family mating structures because it allows us to phase those genotypes and work out the colours of the fragments that are being passed down. The ease of introgression will depend on the genetic architecture, so how many genes there are, whether they are dominant or additive or recessive. And introgression will require some scale. You need quite a lot of animals, and it will require a multiple year commitment. And that's all for me, thanks.
Thanks very much, Dorian. So first up, we do have a question through on Slido. Is Alistair Campbell from Earnscleugh and the Merinos with Myomax and Inverdale an example of grading up?
So I mean, Myomax is a gene that was stolen from Texels if you generated a composite, if you like, and it would be grading up if you went totally back to another breed like a Romney or a Coopworth that had the Myomax that you'd got out of, say, the Texel. Then that would be grading up with introgression. Yep.
Please keep the Slido questions coming through, but we've also got throw boxes here for those not using Slido. First of all, we'll go to Robert.
Is this on? Dorian, I understand that with this sort of crossbreeding, backcrossing, whatever, you never really get truly rid of some of the traits, but
without genomics you never do. With genomics you can if you want to.
So I'm just a bit confused as to why I've got a Romtex flock, I've got a ram that is on paper and agreed with by DNA that he's 20% Texel, 80% Romney, mated him to a ewe that is 60-40, and the progeny, according to DNA, is 100% Romney on cell. Any suggestions
okay,
of
well, I mean,
reasoning?
there's a number of problems. One problem is that if you're using, say, a 50K SNP chip, there's actually millions of variants in the genome, and you're only interrogating 50,000 of them. So there's guesses being made about what the composition of those big fragments are based on just a few SNPs that are in there. Secondly, there's an issue about how good that prediction is. That you see these same kind of things being done for things like dog breeds, where you send in a crossbred animal to tell you how much, Great Dane and Poodle and everything else it is. And that's based on some kind of a training population that is used to dictate how much of each of those breeds there are. And that technology is often somewhat less than perfect. What I'm talking about here is where, say, we're tracking them from the original founders right down, and then we can tell you, pretty much exactly. There's a tiny little window where we might be uncertain about the recombination, but we could very reliably tell you that this fraction of the genome comes from this breed and this fraction of it comes from the other breed. Basically on SNP chips, you're better to ask, I don't even know the technology that is being used for that prediction, but it will be based on some kind of a training population, and it will be limited to the density of the SNP chip that's being used. And so with a 50k SNP chip, it may not be all that reliable. And there's been a lot of work done in beef cattle, say, where they have had very reliable purebred data, and they also find that some breeds they can't distinguish very well. A classic one would be, say, Black Angus versus Red Angus. It's very hard to tell those ones apart. But other breeds, like, say, a Simmental, like a continental breed versus a British breed, you should be pretty good at getting those pieces recognised separately. So direct that question at whoever it is that gave you those percentages.
Thank you.
Oh, thank you, Robert. We've got another question through on Slido. Will gene editing ever happen in New Zealand?
So I have one graduate student that's been doing a bunch of gene editing, involved in a bunch of gene editing, and just published a paper on it in conjunction with AgResearch. And I have other colleagues that are doing a lot of gene editing in New Zealand. But in most of those cases, those animals are never actually being born, or they're never actually being allowed on commercial farms. I would hope that it is allowed, and I would hope that things like the COVID issue would allow people to see that there's benefit from those kind of technologies. But if it's not allowed, they'll end up coming in here anyway, unless we shut the border to importation of everything. And we used to have a pretty tight border, and it was very hard to get things like sheep in. Now we have a very leaky border. It seems to be very easy to bring things in, and it will be very hard to stop things from coming in if they've been gene edited, because there's no way of demonstrating in most cases that they have been gene edited. And I think if we shut the border, we'd probably have people complaining about the WTO violations.
Ross, I think, was next.
Sort of a two-part question, Dorian. So the Massey have got a program just started up with Wiltshires. Is that going to utilise your ingression selection? And if the second part of the question is, if some of us commercial guys wanted to do it, how expensive is it? How do we go about it?
Yep, okay. So Massey has a research project going on at Riverside where they're trying to characterise for farmers what happens if you just go and take Wiltshires and you grade up to Wiltshires. So how many shed in the F1s, how many shed in the F2s, what's the effect on profit, what's the effect on reproduction. It's really characterising the Wiltshire relative to what they've got there already, to just give you an idea of how quickly it will operate. When that study started, then Limestone Downs got interested in doing something similar, and Limestone Downs mated all of their tutus to Wiltshires to generate F1s. And at the moment, that is all that has been funded up there. What I would like to do is go in there and genotype those founder Wiltshire sires and those F1s and the subsequent animals down the line, so we can see where these regions are. I found it very surprising when I looked in the literature how little data was out there on Wiltshires. I thought somebody would have mapped these things already, but that's not the case. So yes, we are very interested in doing it if we can get the funding for it. The bulk of the funding is just the SNP chip analysis. The phasing is a computational exercise which BLG must be doing every time they run a single step analysis, because about 0.1% of SNPs are missing normally, and the statistical methods we use don't allow missing SNPs. So the first stage you do is something called imputation, which fills in the missing ones. And behind the scenes, the way imputation is done is by phasing them. So the phasing is kind of it's just a computational thing that needs to be done, but it's best done when you have family structures in this context. So you would need to have little SIB families where you wouldn't have to know who the sire is. If you had put 10 sires out with your ewes and you had genotyped the 10 sires and you genotyped all those offspring, as long as they were done with something like a 50K chip, we could resolve the parentage and we could phase them from that. And if you can phase the piece that came from their father, then you know that the remainder of their genotype must have come from their mother. So you don't have to have done both sexes all the way through. But it is future in the sense that right now there is no commercial system in place through SIL or BLG to do this kind of thing.